Hello and welcome to this video podcast for human genetics. In this video podcast, we're going to talk about the structure of DNA. And I'd like to begin by just drawing a brief double helix here. The double helix is equal to the two nucleic acid strands. One strand is here and the other strand is wrapped around. As Chargaff's rules helped explain, they're held together by hydrogen bonds between the two different strands. Each nucleotide, guanine, adenine, cytosine, or thymine, will have these hydrogen bonds on either side holding them together. We know that in this DNA, that a portion of it, or several portions of it, represent a gene. And we'll talk about genes later on when we talk about transcription. But these are ultimately what goes on to make proteins. But what I'd like to do is spend a little bit of time talking about what makes up these nucleic acids. What are the smaller individual structures? And as I said before, the individual structures are nucleotides. So nucleotides are the building blocks of nucleic acids. Now your book provides elaborate structures of what each of these nucleotides look like, but I'm going to draw everything that you actually need to know as far as our exams are concerned. Every nucleotide has three parts. So let me list these three parts for you. It has a nitrogenous base. It has a phosphate group. And it has a sugar. So let me draw what this looks like over here. Now, you're never going to have to draw this on the test, but you should be able to recognize it and be able to identify the correct version of it. In DNA, the sugar is deoxyribose. RNA is just ribose, and we'll talk about that later. But deoxyribose, but the key for deoxyribose is there's a hydrogen here. Ribose, as we'll see later on, has a hydroxyl group there. So look for this hydrogen here, and that will identify it as DNA versus RNA. It also has a hydroxyl group over here. This is known as the 3' hydroxyl group because this is the third carbon. And don't worry too much about it, but I do want you to know that that's the 3' hydroxyl group. Okay, attached to this carbon group on the sugar is the phosphate group. This carbon here we call the 5'. And 5' and 3' will become important in just a moment. Now over here is the base. And for our purposes, we're just going to draw it as a square. Now I'm going to spend just a little bit of time talking about these bases, so I'm going to make some space over here so I have that room. There are 5 bases. 3 of them are pyrimidines. They are cytosine, uracil, and thymine. The other 2 bases are purines, and they include guanine and adenine. I should also say we often just refer to them by their first letter. So G and A here and C, U, T here. Now, our DNA only has 4 bases. And so I'm going to put DNA here by cytosine because that's found in DNA, and thymine, and guanine, and adenine. We haven't talked much about RNA yet, but this is an appropriate time to talk about it here, is uracil is not found in DNA, but it's found in RNA. And thymine is not in RNA. But RNA does have cytosine, and it does have guanine, and it does have adenine. As a trick that I've used in the past to remember which ones are pyrimidines and which ones are purines is this. The pyrimidines spell out the word cut, C-U-T. And again, I know this is silly, but this is how I've always remembered it. If you think of a pyramid, the pyramid has a point on the top. And if you use your imagination, you could imagine cutting your finger on this very sharp point on the top of a pyramid. So you could cut your finger on a pyrimidine. Okay, I'm glad I'm not telling you this face to face because I'm sure you're laughing at me at this point here. But I guarantee you some of you will forevermore remember which ones are pyrimidines because of what I just told. The 2 that are left are purines. No fun way to remember that except they're just the 2 left. Now earlier I said that I needed you to remember 5' and 3', and I'm going to explain why now. Okay, I redrew it, but smaller here and with a few less details, because I want to explain what's happening here. And I want to talk about why the 5' and 3' ends are important. Remember that our double helix over here is nothing more than a series of these nucleotides. And they have to be connected to each other. Otherwise, the helix wouldn't stay together, of course. And they're connected right here at this 3' hydroxyl group and the 5' phosphate group of the next nucleic acid. And so I'm going to erase this here, and through a chemical reaction here, it would appear as such. And then the next one would be added here, and so forth. Now remember, that is just one strand. And so as I've been showing with this blue here, maybe this is that strand. And so the very top of it here, and so the very top of this double helix here is that 5' phosphate group. And the very end of it, after many, many more nucleotides have been added, that's going to be that 3' hydroxyl group. Now there's going to be another strand over here. And I'm not going to draw it, but I do want to draw at least the basis of it. Well, I guess we can at least draw it like so. Lacking a lot of detail here, but that's okay. I'm mainly showing you this because this is where those bases come together. So if this is an adenine, this will be a thymine. And if this is a guanine, this would be a cytosine. And as I mentioned before, the A's and the T's, there's 2 bonds, and the G's and the A's, there are 3 hydrogen bonds. Okay, so these are hydrogen bonds. And these here, I should label those, these are called phosphodiester bonds. Phosphodiester bonds are between 2 adjacent nucleotides. The hydrogen bonds link the 2 strands. I just realized that I kind of messed up with showing the strands on this. I'm going to erase this because I won't sleep well unless I show it correctly. For your purposes, if you don't want to redraw it, that's okay. So remember that over here we had this strand, and it's going to wrap around with the 3' down here and the 5' up here. Over here, one thing I didn't explain to you, and it's kind of important for you to understand the principle of it, not necessarily how it happens, but these strands are what we call anti-parallel. What that means is that on this other strand, the 3' hydroxyl group is here, and the 5' phosphate is here. And so what that means for practical purposes over here is that you're going to have the 3' hydroxyl group for this strand here, like so, and all the nucleotides added along here, and it's going to end with this 5' phosphate group here. Big picture, if you're trying to figure out what do I actually have to know from this, so this is what you really should know, and maybe we can make a short list here. You should know that DNA is a double helix. You should know that DNA has 2 strands that are anti-parallel. You should know that the building blocks of DNA are nucleotides. Nucleotides in DNA consist of a phosphate, a deoxyribose sugar, and a nitrogenous base. You should know that those bases in DNA are adenine, thymine, guanine, and cytosine. You should know that thymines bind to adenines with 2 hydrogen bonds, and guanines bind to cytosines with 3 hydrogen bonds. You should know the types of bonds we talked about. You should know hydrogen bonds. You should know that these hydrogen bonds are between the 2 strands of the DNA. So let's put between strands. And you should know phosphodiester bonds. And these are between nucleotides. And I don't have much room here, so I abbreviated between B/W, and then nucleotides is just NT. So remember, those phosphodiester bonds are between the nucleotides on a strand. Of course, you should know that DNA is the genetic material, but that was from the previous whiteboards. Well, I'm out of room for the most part, so I guess we're out of the main points. So if you know these 5 main points and can identify them in a drawing, you will be in very good shape as far as the DNA structure is concerned. Now, looking at your note-taking guide, there's one more term that I want to talk about associated with the DNA, and that is a nucleosome. In a typical human cell, we have a nucleus, and this nucleus contains all of our chromosomes. And remember, A chromosome, if we were to draw it like so, is nothing more than one large piece of DNA, a double helix, a large DNA molecule. That's what a chromosome is. Interestingly, if you took all the DNA from a single cell and put it end to end to end, you would have 2 yards, or approximately 2 meters of DNA. I don't know what you think, but that always amazes me, that inside of this tiny nucleus, the DNA that stretches for 2 yards is pushed in there. And not only is it pushed in there, but it's pushed in there in such a way that it can be used. Think about packing for a vacation. I can cram a lot of stuff into a suitcase. A lot of clothes, all my toiletries, everything I'm going to need for that trip. I can cram a lot in there. But if I'm not careful about how I push all that stuff into my suitcase, when I get to my destination, it's going to be perhaps useless because it's going to be wrinkled. It's going to be maybe the shampoo opened up because I wasn't careful. It's not going to be very useful. But if I'm very careful about how I put my clothes in there, I roll them up tightly. I make sure everything's safe from things that could spill. Then when I get to my destination, they're usable. And that's what I think about when I think about the nucleus. That DNA isn't shoved in there like I would normally pack for a vacation, but it's placed in there in such a way that maybe my wife packed my luggage. And the stuff is now usable. And the DNA in the nucleus is incredibly dynamic and incredibly usable. And it has to be, because we have 23,000 genes in each cell, but only a few of those genes, relatively speaking, are used at any given time. The rest have to be packaged up in the corner to be used at another time, and only the ones that need to be used are exposed. So you might think of our DNA in a cell looking something like this. Now we couldn't really easily put that in here, but what happens is this DNA is tightly packaged, and I'm going to draw it like so, with these proteins around it called histones. That's the name of the protein. This structure of the DNA and the histones, that's called the nucleosome. There's a lot more detail to it than just this, but that's all I want you to know as far as the structure of the nucleosome is concerned. And there are repeating nucleosomes all the way down here, each with those histone proteins in it. These then are folded on themselves even more so that they can be placed in this nucleus. And the reason that these are usable is as follows. Let's pretend that there is a gene under this nucleosome. And we needed to turn that gene on, but we wanted to keep these genes off. It's easy for our cell to remove this nucleosome in such a way that you might have something looking like this, where we still had this part here packaged up in a nucleosome, but the nucleosome that was once packaging this part where our gene is at has been removed. And now our gene here can be used. And when I say used, I mean it can be transcribed and translated into a protein, which then can be used for some function of the cell. And we're going to talk about transcription and translation soon. So the nucleosomes do these things. They package our DNA into usable but compact structures. And nucleosomes help regulate which genes are used. And that's all I want you to know about nucleosomes. On the next whiteboard, what I'm going to talk about is how we take this DNA here and we replicate it during cell division. So let's start talking about DNA replication. And I'm only going to scratch the surface of this. As I begin this discussion, I want to remind us of the cell cycle that we talked about earlier in the semester, where we had our G1 and G2 and our M phase here, and we had our S phase here. DNA replication happens during this S phase. And so that's what we're going to focus on right here. Because earlier we talked about how you might have a cell with 2 chromosomes, and after S phase, that cell's going to look like this, with 2 copies of it. Each one of these strands serves as a template to make another strand. And that's where we're going to focus. Let's draw our DNA like this. Or one strand I'm going to make green, and the other strand I'll make orange. We say that DNA replication is semi-conservative. Your book explains why we know DNA is semi-conservative and not replicated using a different mechanism. You don't need to worry about those experiments. I'm not going to talk about them in this class. But I do want you to know that DNA replication is semi-conservative. And what I mean by that is that the new DNA made conserves half of the original DNA. So during semi-conservative DNA replication, these strands are pulled apart. And I'm going to simplify it by just drawing it as a straight line. And the new DNA is made using each of these as a template. So new DNA will be made using this orange strand as a template. And on this strand, the green strand will be used as the template. And so the new strand, I'm just going to draw that by a dashed line here, showing the new DNA using the orange strand as a template. And same thing here, we have the green strand from over here used as a template to make a new strand. And of course, each of these here will form that double helix. So semi-conservative because the new strands conserve half of the original strand. Half of it. Now I want to go into a little bit more detail about how this is replicated. Okay, I'm going to draw this double helix here, like so. What happens is at a certain region of the DNA, which we'll call the origin of replication. So how are we going to go from this to this? Two new strands of DNA. Well, there are a few enzymes I want you to know. The first is helicase. And what helicase does is it breaks the hydrogen bonds between these two. It's sort of, if you think of it this way, it acts as a saw and saws between these two, breaking all those hydrogen bonds. And what results are regions that are now single strands. The next enzyme I want you to know is an enzyme called DNA polymerase. And DNA polymerase, and I'll draw it in a different color here, will add that new DNA. And so I should draw our end products over here as orange and green too, just to avoid confusion. So sorry about that. I should have planned that out a little better. And the last enzyme I want to talk about, and I'm not going into great detail, but it's an enzyme I want to talk about because it has clinical implications. And that enzyme is called telomerase. We've known about telomerase for a long time, but we still don't understand it as well as we'd like. But what we do know is that it's important in making sure that these ends of the chromosomes get replicated. Without going in detail, DNA polymerase has some limitations, and those limitations are found when it comes to replicating the ends of chromosomes. And if you don't replicate the ends of the chromosomes get shorter with each cell division, which can be good or it can be bad. And I want to talk just a little bit about that. But while we're on this whiteboard, I want to remind you that DNA, as one double helix here, is broken in half using the enzyme helicase. Each strand that results after helicase is used as a template for DNA polymerase to make the new strand that we see over here. And that telomerase is an enzyme that's important in maintaining these ends. That's all I want you to know about DNA replication. So just a very little bit about telomerase and telomeres. So here's our chromosome, and we refer to the ends of the chromosomes as telomeres. Telomerase maintains these ends. However, if no telomerase is present, so no telomerase, the ends cannot be replicated faithfully. And so what results is after one division, they get a little shorter. After another division, they get a little shorter. And at some point, this is going to cause the cell to die. Now, at first thought, that might seem sad. We don't want our cells to die. But it turns out that often we do want our cells to die, because a cell that lives for too long can accumulate mutations, and that can lead to cancer. So this is actually a good thing, that there is this mechanism built into cell division and DNA replication that signals our cells to die. So this prevents the accumulation of mutations and ultimately helps prevent cancer. It's not perfect because cancer still occurs, but it can help prevent cancer. In fact, we know that, and I'll just draw that over here because I don't have room here, that cancer cells often have telomerase expressed. Again, we usually don't want telomerase expressed, because we want these cells to die. But if a cell has become cancerous, often telomerase is expressed. So we'll remove the no there. And now the chromosomes don't shorten after each cell division. And after enough cell divisions, what is going to happen is you're going to accumulate mutations, and maybe more and more as time goes on, which can lead to cancer. So why do we even have this gene present? Why would we ever want to maintain these ends? Because we know maintaining these ends can lead to cancer. Are there any cells that we want this enzyme expressed? So in cells with telomerase expressed, these telomeres will also be maintained, and the chromosomes in these cells never get shorter. Now, as I said a few moments ago, when it's cancer, that's a bad thing. We want the chromosomes to get shorter to prevent cancer. But in some cells, we want telomerase. And what are those cells? I'm going to bring back an old friend of ours, stem cells. We want stem cells to be immortal. If this is a stem cell, we don't want the telomeres to get shorter and ultimately die. We want this stem cell to survive. Like a rock legend, we want it to live forever. Because these stem cells have to replace the cells that are dying or the cells that get damaged. So these stem cells have telomerase expressed in them, and that is a good thing. Okay, that ends this podcast on the parts of DNA and DNA replication. If you have any questions at all, please let me know. If not, I'll see you on the next podcast. Bye for now.